Cell specialization. First, the human body is an amazing thing. It has over 30 trillion cells by current estimates, and over 200 types of cells, each with a different shape, size, and function. But at the same time, all your cells have the same DNA, the same blueprints. So why aren't all cells identical? This is where cell specialization kicks in. Cell specialization is the process by which a less specialized embryonic cell becomes a more functional specialized cell type. A metaphor I like to use for cell specialization is to think of an artist with a palette painting a canvas. The paint by itself does not have a purpose, right? It doesn't evoke any feelings because it's not been specialized on the canvas yet. But once you put it on the canvas, then it can finally serve its function. And this is what cell specialization does. It's the conversion of a more versatile cell into a more functional cell, more specialized cell. To understand exactly what a specialized cell is, we're first going to have to look at how an embryo develops. Of course, you know how life first begins. You start with two gametes, a sperm cell and an egg cell, which together form a zygote with a combination of both their DNAs. The zygote will then divide and form a group of totipotent cells. Totipotent cells are the most versatile of stem cells because not only can they turn into any cell type, such as a nerve cell or cardiovascular cell, they can also form a whole new embryo, a new organism. In fact, in nature, when a totipotent cell group spontaneously splits, both groups will eventually become an identical twin. After totipotent cells divide, a hollow sphere of cells called a blastocyte is formed. Inside the sphere are a cluster of inner mast cells, our second type of stem cell. This new type of stem cell will grow on to form the embryo, while the sphere around it will form the placenta the embryo needs to survive. These inner mast cells, or pluripotent cells, can turn into any cell type, but unlike the other stem cells, totipotent cells, they cannot form new organisms, new embryos. Pluripotent cells are the cells used in embryonic research, but such research is still under debate. After the blastocyte is formed, an embryo finally begins to develop, and the cells are specialized. These are the more traditional type of cells. Your skin cells, your heart cells, your liver cells, all those cells in your body with a function or specialization, and they are called unipotent cells. Sadly, once you go unipotent, you can't go back. Unipotent cells can't revert back to stem cells. You can't have a blood cell turn into a stem cell, turn into a bone cell. That's not going to happen. Most cell types will remain the type they are. So I've told you the steps to cell specialization and the types of stem cells, but I haven't explained yet what actually happens in the cell. Each cell is controlled and told what to do by DNA. But since cells all have the same DNA, the same set of instructions within an organism, nature has to come up with a way so that every cell type has the instructions for their specific function only. Nature accomplishes this by giving every cell the instructions for every other cell as well. For example, in the DNA of a nerve cell, only a small part of the DNA is expressed. The rest is tightly wrapped so that it can't be read or transcribed by RNA. For example, casein is a protein found in milk. All cells contain a casein gene, but only milk-producing cells express that gene. In other cells, it remains silent so that casein is not produced. But the silencing and expression of genes is not permanent. Gene regulation changes constantly. Various stimuli will cause proteins called transcription factors to promote or even block gene expression. And that's it. You should have learned what cell specialization is, the type of stem cells, how they develop, and how transcription factor works.